Okay, so we're on to our final video on digestion. So the last part of what we're going to do is we're going to talk about lipids. So we're going to talk, like always, about structure first. And you're going to notice the structure is a little bit of a different story this time. We're still going to break large molecules down into smaller molecules, but we're not going to have as much of a story of chains of molecules. So we primarily consume our lipids in three different forms. One is triglycerides or triacylglycerols. So it's a glycerol backbone with three fatty acids attached to it. So this one's our most significant. We consume cholesterol, and primarily a cholesterol ester. And we'll talk about that a little bit more. We also consume some phospholipids. So we can group all these together as polymers, and we can talk about what we're going to break them down as or into. So let's move up a little bit here. So the triglyceride, and let's talk about the structure of that for a second, and then I'm going to go back to abbreviating it as a TAG. So what we have there is we have a glycerol backbone. It's the 3-carbon alcohol glycerol, or that's what it starts out as. And we attach three fatty acids to it. We do them through an ester bond. And remember when we were talking about terminology, we said, GL anything, of course, you're going to think carbs. Am or pep, you're going to think proteins, polypeptides. Ester, anything, lip anything, or acyl anything. You're going to be thinking about lipids, fats. So ester bond, that's where your mind's going to go. And of course, our ester bond is the carbon double bonded to one oxygen, single bonded to another, and attached to something else on either side. So this part started out as our glycerol. We've got our ester bonds attaching some hydrocarbons, or when we made them, what was fatty acids. So this structure altogether with the three fatty acids attached to it is a triglyceride, also known as a triacylglycerol, also known as a TAG. So we're going to talk about that structure and play with it a little bit here and there, but let's talk for a second about something that's kind of crazy obvious, but sometimes when you're drowning in the details of enzymes and hormones and everything, it's easy to miss. When I have all three hydrocarbon chains on there, it's a triacylglycerol. If I have two of them, it's a di. One of them, it's a mono. So we're using TAG as our abbreviation for the tri. We're not really going to do anything with a di, but when we do, we'll use DAG. For a mono, we're going to use MAG. Then for a free fatty acid, we'll either do FFA or FA. So you're going to see that terminology or those abbreviations as we go through, kind of to save space on the screen a little bit too. So let's continue our conversation about structure and what we're going to break everything down into. So again, we have the polymers, triacylglycerols, cholesterol esters, and our phospholipids. So our triacylglycerols, we're going to break those down into mono. Remember, we took two fatty acids off, plus some fatty acids. We're going to take our cholesterol esters, which are kind of just a cholesterol with a fatty acid attached to them, and we'll get cholesterol out of that. We're going to take our phospholipids, and their structure is awfully similar to the triacylglycerol we just drew. We just put a phosphate in between. So before we attach the hydrocarbon, there's a phosphate there. We leave it. So we talked about the structure a little bit, but just to remind you, that's going to give us some glycerol and some fatty acids. So these are all the monomers that we're going to break those polymers down into. Again, a catabolic process like we used for breaking down our carbs, breaking down our fats, catabolizing the larger molecule into the individual monomers. You'll notice a lot of similarities there. So we're going to start this story in the mouth, in the oral cavity. So we'll just say mouth. It's easier, but we really mean oral cavity. And we're going to say you ate some fat. So we were having still our ham sandwich, and we had some nice cheese on there, maybe some mayonnaise. Maybe we went Canadian, buttered the bread first. But we had some fatty stuff on there. So there's a couple of glands in the oral cavity that secrete an enzyme that we need. So we have the sublingual salivary glands. We have the parotid salivary gland. And what they do is they're going to secrete lingual lipase. So we'll get our lingual lipase. What that's going to start working on is it's going to start working on our triacylglycerol. So I'm going to get very abbreviated in how we're going to draw that. So we're going to draw the 3 carbons for the glycerol backbone. We're going to draw the fatty acids, just Fa, because we went over the structure. And what our lingual lipase is going to do is it's going to break off 2 of the fatty acids. So we'll just have OHs, hydroxyls on there. So what we end up doing is we take our triglyceride, convert it to a monoacylglycerol, remember 1 left on there, and we're going to get 2 free fatty acids. So that's our first step. That's what our lingual lipase is going to do. And we can't break all of the ester bonds. And the reason for that is kind of obvious when you think about it. The food moves through your mouth pretty quickly. You put your food in your mouth, chew it, swallow it, and move on. So we don't have a lot of time to break down a lot of stuff in the mouth. We're going to have a bile story later, which will be a story of more stuff getting broken down. So our next stop is going to be the stomach for a second. So in the stomach, again, we're going to have chief cells. And along with the pepsin that the chief cells released, the chief cell is going to release gastric lipase. So we're going to continue breaking ester bonds there. So again, we'll do a little bit of work in the stomach. We'll release the gastric lipase, a little bit of work, but the main part of our work, like was true with most of our digestion, happens in the small intestine. So next stop is going to be the small intestine. And the bulk of our story is going to happen here. So we said one difference with breaking down fats as opposed to our carbs or a protein was that we have less of a long chain kind of story getting broken into smaller pieces. The other big difference is fats are not water soluble. We have the oil and water story going on. So we're going to have another part of the story because of that. We need bile, we need some other molecules to get this non-water soluble molecule broken down in a relatively aqueous environment. So the duodenum, again, the first foot or so of the small intestine, is going to release a couple things. So it's going to release bile, and we'll talk about what that does in a minute and how it does that. It's going to release pancreatic lipase. So you see in the lipase story kind of continuing through. So let's back up and talk a little bit about the bile story. We have the gallbladder. And the gallbladder's job is to store and concentrate bile. So bile is the gallbladder's department. We have the liver. And we also have the pancreas. So we kind of have multiple players in this story. So we have the right and left hepatic ducts coming out of the liver. We have everything coming into the cystic duct. And we have the pancreas with the main pancreatic duct. We have, remember, with our pancreas, we have the sphincter of Oddi. So it's smooth muscle coming out of the pancreas or the hepatic pancreatic ampulla, whichever word you want to use, ampulla or vater. So what we have is smooth muscle there. And what's going to happen is we're going to release bile from the duodenum. The liver is going to secrete the bile. The gallbladder is going to store and concentrate it. Then we have the pancreas adding some enzymes to the mix. So what we're going to do with all of this is break a large fat droplet down into some smaller droplets. So what I'm really talking about here is a process called emulsification. And it's kind of a surfactant process. So the best way to think about it is, let's say you're rinsing the dishes off and you're going to put the dishes in the dishwasher. So, carb-based stuff rinses off, protein stuff, you don't even notice it rinses off, but you start running the water on a plate that has some fatty stuff on it, butter and stuff like that. And the butter kind of rolls, or the water kind of rolls off of it. So what you do then, take some dish detergent or whatever, throw that on. And if you really look, it kind of breaks it up. And now we can rinse the dish off. So that breaking up process is emulsification. That's the job of the bile and the bile salts. So let's break that down a little bit and talk about how we're going to do that. So we're still in the duodenum, and the bile is made up of two things. Bile is made up of some phospholipids, and we'll talk about their job. And that's things like lecithin, stuff like that. The bile is also made up of bile salts. Cholic acid, derivatives of cholesterol. They're going to react with taurine, glycine, things like that. So what I have is the fat globule wants to be a globule. Fat wants to kind of stick together, get out of the water, that whole hydrophobic, hydrophilic thing. So I have this fat globule. I've got some triglycerides in it. I have some cholesterol esters in it. I have some phospholipids in it. And I have that in this big globule. And it's floating around in the intestinal chyme. And the chyme is a watery kind of solution. So I have pancreatic lipase hanging around. What pancreatic lipase would like to do is get in there and start breaking these molecules down. The only problem is because of this big fat globule, it doesn't really have enough surface area to work. So we're going to go through some steps and talk about how we're going to break this fat globule down into smaller particles. So like in the carbon protein story, we had the chain go into smaller chains, smaller chains, smaller chains, finally down to pieces. We're going to have the fat globule go to smaller globules, smaller blobs, and smaller and smaller. So first molecule we're going to talk about working on it is going to be a phospholipid. That color didn't work very well. I'll have a green phospholipid. And the thing with the phospholipid is it has a hydrophobic part and a hydrophilic part. That allows it to interact with the fat globule. So I've got a hydrophilic side. Of course, the hydrophilic side likes being in the water. I've got a hydrophobic side. So that wants to interact with the fat. So with this, what we're able to do is we're able to break this globule down into smaller fat particles, I guess is fat droplets, probably a good term. So what'll happen, we'll break it into the smaller droplets, and that phospholipid is going to stay around it. So now I've got my smaller droplets. So we're making progress. So I've got my smaller droplets. What color did I use? Blue droplets with green phospholipids. Now the bile salts are going to come in. So the bile salts have the hydrophilic, hydrophobic part. We have the negative charge on one side. So with the bile salts, the lecithin is going to come and join the party. The bile salts will come in, what color can we use? I guess we'll have to go back to blue. We've got the bile salts around the particle. Then, once we have all that around there, we have another protein that wants to bind. So another protein wants to bind, and it's a protein called colipase. I think I'll use my funny purple for that. And what colipase does is it's going to help the pancreatic lipase bind. So when we get the pancreatic lipase bound on there, that's going to break down some more triacylglycerols. FFA for free fatty acids. So this is the job of the pancreatic lipase. So we're getting broken down a little bit more, and this was the bile salts doing that. So now we have two other chemicals that are in the pancreatic juice. We're going to have, I guess we need a new slide for this. So we have two other chemicals. We're going to have phospholipase A2. PLA2. We're going to have cholesterol ester hydrolase. And remember, cholesterol ester is really just a cholesterol with a fatty acid bound to it. These two are from the pancreas. So what we're going to end up with is the phospholipase A2 breaks down the phospholipid, gives us glycerol plus free fatty acids. So you can kind of see a theme here of what we're getting. The cholesterol ester hydrolase adds water, breaks down the cholesterol ester to the cholesterol and the fatty acid. So we're getting all these smaller pieces again. So now we have what's called micelles. Let's spell that. And they're tiny droplets, and they're made up of all these things. Still have the phospholipids, still have the bile salts attached to them. They're also going to pick up, so they have the phospholipids, they have the bile salts. I won't try drawing them. They're also going to pick up some vitamins, A, D, K, and E. Well, of course, they're the fat-soluble vitamins. Now, with all that, we can go to the usual place. We can go to the enterocyte. What's interesting, though, all of this stuff, or most of this stuff, and all of the stuff that we just made on previous slides, this is all going to go to the enterocyte, except for some of the bile salts. Some of the bile salts can go back to the liver and get recycled. So all the other stuff then can go to the enterocyte. So let's do what we've been doing all along and draw our little enterocyte and talk about what happens where. So I don't know if we have a color theme with our enterocyte. I think we did blue. So we have our enterocyte. And we have some stuff. We have our cholesterol. We have our vitamins, our fat-soluble vitamins, A, D, K, and E. We have some free fatty acids. We'll do FFA. We have some monoacyl glycerols. So they're going to go into the enterocyte, just passive diffusion. So they'll go into the enterocyte and they're in there. So once they get in there, we're going to talk about what's happening in a couple of parts of the cell. So first part we're going to talk about is the smooth endoplasmic reticulum. And all of these are going to go to the smooth endoplasmic reticulum. So some of the cholesterol go back to the ester form. We're going to put some of the tags back together in the smooth endoplasmic reticulum. So what they're going to do is we're going to package all this stuff together. The cholesterol, the vitamins, the free fatty acids, the monoacyl glycerols, Some cholesterol go back to the ester form. We'll actually put some triacylglycerols back together. Get it all packaged up. Then it's going to go to the rough endoplasmic reticulum. So we'll take that, ship it off to the rough endoplasmic reticulum. So what we're doing in the rough endoplasmic reticulum is we're taking that little package that we have. We have cholesterol in there. We have cholesterol esters in there. We have the vitamins in there. We have phospholipids in there, triacylglycerols, free fatty acids, all of that. What we're going to do here is we're going to put a nice little protein coating on it. The little protein coating that we're going to put on it, that outer protein, is ApoB48. We're going to talk about lipid transport in another video, but what we just formed was, we'll stick with green, is we formed a chylomicron. So we packaged everything up in this nice little package, and we put this protein coating on it. What we're going to do with that, which is different, rather than shipping that off to the bloodstream, it's going to go off to the lymphatic capillaries. So it's going to go off to lacteals. So it's going to travel through the lymphatics to the thoracic duct. And it can go to different organs. And we'll really pick up this story more when we're talking about lipid transport. So some of the different organs it can go to skeletal muscle, it can go to adipose tissue, and some of it will go there. Some of it's going to go back to the liver. When we go back to the liver, we're going to start picking up more proteins, things like that, make our VLDL, ship it back out. So at this point, we'll probably stop the fatty or the lipid story, pick it up in another video talking about how the chylomicron becomes VLDL, dropping off fats, picking up proteins, and things like that. So we will stop there.